Welcome to chapter 12, and this is the third part. So we finished talking about the sodium and potassium changing places. We've talked about the various types of nerves in the central nervous system versus those that are in the peripheral nervous system. We've talked about Schwann cells, oligodendrocytes. So now we're going to get down to the nitty-gritty, and we're going to actually look at how the axon talks to the postsynaptic membrane. So in this particular picture, this is an actual picture taken with an electron microscope, and what you see here are the ends of axons. So we talk about the axon coming down, and then we talk about it branching out into little terminals, and at the end of the terminals are little boutons, which are the little buttons at the ends of the terminal, and they have the vesicles full of neurotransmitter. So we went over this in the chapter on muscles, so some of this is a review. Now, you have basically three things that the axon can talk to. It can talk to another nerve, it can talk to a gland and cause it to secrete, or it can talk to a muscle and cause the muscle to contract. So in this particular picture, what we're looking at is an axon. So here's your axon. There is the axon terminal, the bouton. Here they all are. But what it is making a synapse with is another neuron. So this is the cell body, the soma of a post-synaptic neuron. So the pre-synaptic is this axon coming in, and it's going to be delivering neurotransmitter across the synapse right here, and it's going to be stimulating this neuron, this nerve cell right here. So again, you can stimulate a nerve cell, you can stimulate a gland, or you can stimulate a muscle. A couple of things we're going to talk about before we finish this chapter is how does a neurotransmitter work? What is a neuromodulator? What is its actions? And how do you stop the stimulation of a postsynaptic cell? So you've stimulated the muscle, the nerve, or the gland, but now I don't want to stimulate anymore. How do I stop it? How do I stop the neurotransmitter? In our last lecture, we talked about how you get the action potential going, and you have the sodium potassium changing places across the phospholipid bilayer. But what we actually are doing is we're transmitting electricity. So that millivolt thing, negative 70 millivolt, going up above 0 millivolts, then going back down below negative 70 millivolts, that's electricity. It's not electricity like what you get when you plug something into the wall, but we're talking about something that you need a microscope to see. So the electricity only goes to the end of the axon. Once it reaches the end of the axon, that's the end of the line. When it hits the bouton, it can't go any further. So if the only way that you're going to carry the message any further is you're going to use the electrical action potential to release a neurotransmitter. And when the neurotransmitter goes across the synapse, then it's going to cause a new wave of electrical activity in the next cell. So remember, there's a synapse because you don't want two nerves touching each other, because if they did, they'd short each other out. So the first neuron is called a pre-synaptic because it's on the first side of the synapse, and then whatever is on the other side is called post-synaptic. So that's not too hard to remember. And when I said that you could have a pre-synaptic axon talking to either a muscle, a nerve, or a gland, on the nerve you could talk to the dendrite, to the actual cell body of the neuron, or to another axon. So you can talk to any of the parts of a neuron. And any of these can be the post-synaptic membrane. Now, one of the things that I hope you saw in the picture that we started out with is a neuron can have an enormous number of synapses. So it can be receiving messages from all sorts of cells. Almost all of those synapses will be made with neurons, other neurons. But remember, there's some neuroglia out there that can also make synapses. So they just give you some numbers here. You don't have to memorize these numbers. But the spinal motor neurons, so remember this is coming out of the spinal cord. Motor neuron is the efferent. It's going to cause an effect. They may have up to 10,000 axon terminals touching it. Of those 10,000, most of them are going to end up on dendrites, and about 2,000 of them will end up on the cell body itself, on the neurosoma. In the cerebellum of the brain, you can have up to 100,000 synapses. So someone somewhere sat and counted all of these synapses. So it was exactly 100 years from now, before now. Let me try that again. 100 years ago that they found out that there is some sort of a substance, they didn't know what it was, that caused the muscles to contract. So what they did is they took the hearts of 2 frogs and sprinkled saline on them. You just drip the saline across it to keep it nice and moist. And then they stimulated the vagus nerve of the first frog, and it made its heart slow. Then they took the saline away from the first frog and put it on the next frog, and it slowed the second frog's heart, even though you hadn't messed with its vagus nerve. So they figured out, well, there must be some vagus stuff that's coming out from the vagus nerve that is affecting the heart. It's the muscle of the heart. And so they named it vagus stuff. Later on, we renamed it acetylcholine, and it was the first of the known neurotransmitters. So only within the last 100 years have we even known about neurotransmitters. A quick review of what we talked about when we did the muscles. You have the presynaptic neuron. It has synaptic vesicles. Inside the vesicles is neurotransmitter. The post-synaptic membrane has neurotransmitter receptors. So if I put a neurotransmitter in the synapse and there wasn't a receptor on the post-synaptic membrane, then it's not going to do anything. It's just going to sit there. So you actually have to have a receptor sticking out there that is specific for that particular neurotransmitter. And if it fits, if the neurotransmitter gets onto the receptor and changes its shape, then it's going to open up ion gates. And remember, when you open the gates, the sodium rushes in and the potassium rushes out. So you saw it in words, and now we're going to see it again in a little picture form. So you're going to need a lot of ATP energy. So you've got lots and lots of mitochondria. You have the microtubules of the cytoskeleton. So it's pushing things around inside of there. And then there's the vesicles full of neurotransmitter. And they can fuse with the pre-synaptic membrane. And by exocytosis, you can release the neurotransmitter. If there is a suitable receptor on the post-synaptic membrane, then it's going to bind and you're going to get an action potential. You're going to get the sodium potassium changing places. So here it is as a picture, and then you just saw it as words. And just in case you didn't get it, here it is as a cartoon. Because of the action potential or the electricity coming down here to the bouton, you're going to have the vesicles releasing neurotransmitter, tossing it across the synaptic cleft, where it is going to be caught by the receptors on the post-synaptic membrane. Now, there's over 100 neurotransmitters that we've identified, so you guys are only going to learn a handful of those and a small handful of that. So acetylcholine is really important because it's one of the main neurotransmitters that keeps our skeletal muscles going. And it's just as an interesting thing, acetylcholine is formed from acetic acid, which is vinegar. So that's kind of interesting. And your amino acids, you have 20 amino acids. We discovered that your amino acids can act as a neurotransmitter. So they weren't thinking about that when they made NutraSweet because they put 2 amino acids together, and you drink a diet drink that is absolutely full of 2 amino acids that they use to make the NutraSweet, the aspartame. And one of them is phenylalanine, so people who have PKU can't tolerate anything with aspartame in it because they're going to get a mega dose. But what the studies have found is if you drink a lot of this diet sugar, the aspartame, that it will knock out your short-term memory. So I've had students who will be studying for a test and they come in and they go, oh, Ms. Drake, I read this, I knew it, I stayed up all night. I said, did you drink diet whatever while you were doing it? Yeah, I did. And I said, yeah, pretty much everything that you tried to cram in, first of all, you were tired, and secondly, you were knocking out your short-term memory. So if you hadn't already learned this and understood it fully before you started drinking that diet drink, it probably just got wiped right out of your mind. And that's why that you're not remembering it right now. So take-home message there, don't drink something with a neurotransmitter in it right before you take a test. And then we're going to talk about some of the neuropeptides that have the ability to get into the synapse and cause depolarization in the post-synaptic membrane. So here are some of the things that can act as a neurotransmitter. Caffeine, adrenaline, which should technically be called epinephrine, alcohol, now specifically ethanol, chocolate, dopamine, and serotonin. Now, I will tell you that most people say oxytocin is the love hormone. But when you talk about neurotransmitters, dopamine is the love neurotransmitter. Look in this list of some of the other neurotransmitters. Look at that. We see histamine, ATP, serotonin. So in this list, we have amino acids themselves that act as neurotransmitters. We can take some of these amino acids and modify them, and these are synthesized from amino acids. They start out as amino acids, and you change them a little bit, and that's how you make your epinephrine, your adrenaline, your norepinephrine, or noradrenaline, and your dopamine. It also, you have where you can daisy chain the amino acids. So you can put 2 of them together, which we talked about in the case of NutraSweet, and you can make a neuropeptide that works as a neurotransmitter. But you can go up to 40 amino acids. So we have all different lengths of neuropeptides. And they named just a couple of the neuropeptides. Cholecystokinin and substance P are 2 of them. 2 of the really important neurotransmitters, serotonin and dopamine, it says technically these are the only two things you enjoy. So if you feel happy all the time, you may have a lot of extra serotonin or extra dopamine that makes you feel happy. And if you feel sad, then they may give you something that will keep the serotonin from leaving the synapse. So once you put the serotonin into the synapse, it'll keep it there. So they talk about serotonin reuptake inhibitors. So there's a class of medicines out there that do that, and so it makes you feel happier. And I always try to get you to watch other things outside of my lectures that you may find a lot more exciting than my lectures. And one of them is Robin Williams in the movie Awakening. And it's based on a true story. It is truly amazing. There are people who go into end-stage Parkinson's disease quickly. Some of them, we don't know how it happens. It just happens to kids. It happens to adults. And we don't really know how it happens. In one case, it was because there was a group of people who took shots of bad heroin, and it threw them into end-stage Parkinson's disease. And basically they froze, and they couldn't move. And in the movie, Robin Williams portrays the guy who figured out that if you give these people who are frozen a massive amount of dopamine, it'll get into the synapses and it will wake them up. So it was a fascinating movie. I'm not going to tell you how it ends, but if you have the time, Awakenings by starring Robin Williams, based on a true story. It really happened. If you were taking a biochemistry class or if you were taking neuroanatomy, you would definitely be learning all of these chemical compounds. But here's a slide that just kind of shows you some of what the structure looks like of these various amino acids, acetylcholine, the monoamines, and the neuropeptides. Now, I want to show you the enkephalins and the endorphins because these are the things responsible for runner's high, and they're like natural opiates that you make in your body. So that's why you can get addicted to opioids. I think you've probably heard about the opioid crisis, but you can make your own in your body by hurting yourself. So these are released to be your own painkiller that you make. And it is so strong that if you make a lot of this, you'll actually stop eating. Because you don't have time. You just want to be making some more of this so you can keep this feeling going. So it can be dangerous. And they even have cartoons about that. So here's somebody, this normal people, they kind of like each other, and that's what turns them on. And people who work out a lot prefer their exercise because they get more of a high than they would if they were with someone. You've probably heard of people having laughing gas at the dentist. That would be nitrous oxide. But nitric oxide, it can also act as a neurotransmitter. So it can get in and interact with the synapse. Carbon monoxide can also. One of the things that people who sit in their car while they're waiting for it to warm up, and they're in the garage, so the garage door is closed because they want to keep the car warm as long as they possibly can. They're building up carbon monoxide in the car, and sometimes they get enough carbon monoxide that they fall asleep and they wake up dead or don't wake up. Since these are gases, you don't have to put them in a vesicle, and you don't have to do exocytosis out of the end of the pre-synaptic membrane. It can just go freely out and across and over. And it doesn't even need to bind to the membrane surface receptor. It can just go right on in. There are inhibitory synapses, but pretty much the only one that we cover in a class like this would be the inhibitory GABA, G-A-B-A, GABA-aminobutyric acid. The others are going to be some sort of excitatory neurotransmitter. So here we are putting it all together, and we're showing you the exocytosis, all of the stuff. But one thing we're adding in here is you can have an enzyme that can break down the acetylcholine. So that would be acetylcholine esterase, and it will take the acetic acid, the acetate portion, and the choline apart. And once it's done that, then it will not sit on this post-synaptic membrane. It won't allow the sodium potassium to be rushing back and forth across the membrane anymore. So it stops the chemical reaction that's occurring. And then because it's stopping the chemical reaction, it's going to stop the action potential from going down the postsynaptic neuron. So here's your presynaptic neuron. There is your acetylcholine being released into the synapse. Here it is causing an action potential, and because of this enzyme, it's going to break up the acetylcholine, and now this is going to stop. So there's a couple of things you can do to stop the reaction that's going on in the synapse. One of them is to use an enzyme and break down the neurotransmitter. The other one is to just take whatever is in the synapse and put it back up inside the presynaptic membrane. And then you can release it again when you need it, like the serotonin uptake. And here's a slide showing you what GABA is. It is gamma-aminobutyric acid. And its thing, when it is released into the synapse, is it will hyperpolarize the postsynaptic membrane, so it is less likely to fire. So that's why we call it an inhibitory. So when we looked at a synapse that had acetylcholine, we saw where the acetylcholine was released from a vesicle, went across, and it bound to a receptor. And so that was called a ligand-gated receptor. So something bound to it, it changed the gate and allowed the sodium potassium to change places. There are other ways to react with the postsynaptic membrane, and one of them is done with norepinephrine, which is also known as noradrenaline. Instead of this neurotransmitter going in, it's going to bind to a G protein. Now, I don't know if you remember back when we talked about the cell membrane and we talked about all the things that were going on across the cell membrane, the phospholipid bilayer. We talked about G proteins, and it is extending through the phospholipid bilayer, and inside you're going to start a cascade. You're going to activate a second messenger system. So the first messenger is going to be the noradrenaline, and it's going to be binding to this G protein. And then that causes a conformational change in the G protein. It activates enzymes. It activates the second messenger system. And cyclic AMP is one example. That's cyclic AMP. So remember, we have ATP with 3 phosphates, and this has got 1 phosphate, adenosine monophosphate, because you pull 2 of the phosphates off. And when you do this, this is going to take a little longer. So it's not like the sodium potassium rushing in and rushing out and changing the electrical conductivity across the membrane. This one is going to have to bind to the G protein. The G protein is going to have to do its thing and dock with other proteins. And then inside the cell, whatever the postsynaptic membrane is, inside that cell, you're going to make cyclic AMP. And then this is going to cause another enzyme cascade to occur. And this is called amplification. So one molecule docking with one G protein can end up with literally millions of products. So you amplify it through the cell membrane and into the cell. So it's a second messenger system. So it's not the first messenger that does the trick. It's the second messenger inside the cell that causes this big amplification. So here's your little picture showing some of the things that you can do when you dock the G protein. You can turn on an enzyme that this used to be called adenyl cyclase, and then they call it adenylyl cyclase, and now they're calling it adenylate cyclase. So they keep changing the name. But basically, it is an enzyme that will pull two of the phosphates off and take the remaining adenosine monophosphate and turn it into a cyclic compound. So that's why they call it cyclic AMP. And it can activate other enzymes. It can cause genetic transcription. It can cause enzyme synthesis, metabolic changes, or it can do the old going over and do ligand-gated channel openings, like the other thing did, the acetylcholine did. And then you get the sodium and potassium changing places, and you get the electric charge. So there are multiple effects, but this is just an overall. So you can have ligated channels, but you can also have enzyme amplification inside. What's really fascinating is how quickly this takes place. So if you take 1 second and divide it into 1,000 pieces, it only takes 1,000th of a second for the neurotransmitter to cause an effect at the receptor. So that's 1 millisecond, or 1,000th of a second. If you want to end the signal, you've got to get the neurotransmitter out of the synapse. Otherwise, it will keep on coming down, touching the receptor, leaving, coming down, touching a receptor, leaving, coming down, touching a receptor. So you got to get it out of there. So you're going to either enzymatically break it down, or you're going to reuptake. And just in case you forget, serotonin and dopamine are the ones that make you feel happy. A third way, besides using an enzyme to break down the neurotransmitter or reuptaking whatever it is back up into the presynaptic membrane, you can just wait for the neurotransmitter to just diffuse out of the synapse and into the extracellular area. So neurotransmitters can work within a millisecond, 1,000th of a second. But neuromodulators take a little bit longer. So if you have nitric oxide, it's going to help you relax your smooth muscle. So that's just one of the things that it does. And I mentioned the enkephalins and the endorphins that are neuropeptides. You get them with runner's high. So when you start running and hurting, your body's going to start making these, and these inhibit pain signals in your central nervous system. So they stop telling you that you hurt, so that you can continue on doing whatever it is that you're doing. Most of my life, I've alternated between teaching and between doing research in laboratories. So one of the things that I was curious about was you don't have receptors for morphine in your body because you make morphine. That's something that you derive from. Why would we have receptors in our body for a plant? I mean, that doesn't make any sense. So I decided to research it, and at the time I was working on prolactin and its effect on aging. Of course, we know about prolactin because it helps pregnant women make milk, So it's involved in that process. And I noticed that people who are pregnant, who are lactating, people who have tumors that hypersecrete prolactin don't get pain relief from morphine. And so I had postulated that I wondered if the morphine receptor was actually the prolactin receptor. And if you take morphine, it goes onto the prolactin receptor and gives you the pain relief. But if you're already making prolactin and it's in the prolactin receptor, then the morphine has no place to go and you're not going to get any relief from the morphine. And as I did the research, I found out that that was actually true. So all of these women who are breastfeeding their children and they go to the dentist or they go get surgery or whatever, and they give them morphine, they're not going to get the pain relief because they've already got prolactin tied up on the prolactin receptors. So people who have elevated prolactin need to be able to make more of these pain-relieving neuropeptides since they're not going to get the relief from the morphine that most people would. So now that we have looked at how synapses work, how action potentials work, things like that, now let's look at some of the fun stuff. So you know that your brain, you know that the neurons in your brain and possibly the neuroglia are what allow you to memorize, to learn, to recall, to have memories, to make decisions, process, store information. How in the world do we do that? So one of the things that we've been looking at is where in the brain do we store various things? And I'm going to give you a link that I recommend you watch because one of my students gave it to me and I thought, oh my goodness, I'm definitely passing this on. This is a segment on YouTube and it is part of a 60 Minutes where this lady, Leslie Stahl, is talking about mind reading using an MRI machine. So we all know about MRI machines. We know it can image your brain. But what they have now is functional MRI, fMRI, and it actually lights up the part of the brain you're using when you're thinking about a certain thing. And they've been able to map that. And if you think about it, there's only a handful of things that you really think about. Everybody knows what a hammer is. Everybody knows what an ice cream cone is. Things like that. We kind of have a mental picture. But what we don't know is we all store those thoughts in about the same place.
First time in human history to peer directly into the brain to read out the physical makeup of our thoughts. Some would say to read our minds.
History, we could never actually coerce someone to reveal information. Torture doesn't work that well. Persuasion doesn't work that well. The right to keep one's thoughts locked up in their brain is amongst the most fundamental rights of being human.
You're saying that if someone can read my intentions, we have to talk about who might in the future be able to do that.
Absolutely. Whether we're going to let the state do it or whether we're going to let me do it. I have 2 teenage daughters. I come home one day and my car is dented and both of them say they didn't do it. Am I going to be allowed to drag them off to the local brain imaging lie detection company and get them put in a scanner? We don't know.
But before we've even started the debate,
We've looked at individual neurons and what they stimulate, but be aware that usually neurons don't work independently of each other. They usually work in large groups. So when you think about controlling your breathing, think of all the nerves that are necessary, all the muscles that are involved in just breathing. And then when you walk, think of all the muscles that you have to talk to, that you have to innervate. You have to send the message down. So you've got some in your upper legs, you've got some in your lower legs, and then we usually swing our arms to help us maintain our center of gravity. So we have big neural pools and we have nerve circuits. So we're going to be talking about nerve circuits in the next couple of chapters. One of the arguments that I have heard from students over many decades is, well, I have to watch TV when I'm reading my book or when I'm studying or something of that nature. Of course, nowadays they say things like, I can be on Facebook and I can be talking with my friends on Facebook and I can still listen to a lecture. I can still study. I can still be reading my textbook at the same time. Well, yeah, you can't. You can't. You can't read a book and watch a television movie at the same time. You just can't do it. You can go back and forth, but you'll only half understand each of them. So, those of you who sit and are trying to listen to this lecture, and you sit there and you've got your cell phone and you're texting your buddies and you're checking your Facebook, and then when you take the test, you're like, man, I listened to the video and I just didn't get it. It just didn't sink in. But yet, put the other stuff away, because you can't do both at the same time. I don't know if you're like me and you're out driving and you suddenly look around and you think, how did I get here? And you think of all the turns you had to make, all the mergings into traffic that you had to do, and you think, oh my gosh, how did I do this without hitting anybody? Because I don't remember seeing any of this. And we have found that there's a part of the brain that works even if other parts of the brain doesn't work. So here's just a short clip out of a YouTube video called Blindsight.
Simulates a phenomenon called unconscious vision or blindsight that can occur when there's damage to the brain's visual cortex.
So the world to them looks like this. They're blind in the left side of the visual field. And they go to the doctor and the doctor says, well, can you see this? No. Well, can you guess whether it moves to the left or to the right? And the patient says, well, I'm blind. And the doctor says, well, guess. And the patient guesses left, right, up, down. And the patient is as surprised as the doctor, at least they were at first.
Obviously, there are some parts of your brain that you can use simultaneously. You can see someone put an apple pie up on the windowsill. You can smell it. And you can have, it evokes memories of when you were a young child and you waited for that moment when the pie was cool enough that you could eat it. So you can process parallel information. And you have three kinds of memory. You have the immediate memory, where you can remember something briefly, just briefly. And then you have short-term memory, where you can remember it oftentimes long enough to sit down and write it down, or something of that nature. And then, of course, you've got long-term memory, where you remember your childhood, you remember your parents, things like that. So your name, your children's names. So it takes something like Alzheimer's to knock out this memory. And even then, it's one of the last to go. So a lot of times, you can talk to someone with Alzheimer's, they don't know what they just ate, or they don't even know if they just ate. But they can tell you about when they were 6 years old and they got their first bicycle. They can remember that. So, and we talked about short-term memory. So that's where you're trying to put all the information you need in before you take a test. And you're just going over it and hoping that some of it's going to stick. And then we talked about if you drink something with NutraSweet in it, that it will knock out your short-term memory. And so it'll be gone. If you're lucky, short-term memory may last a few hours. Now, there are some people who believe that even the stuff that you put in your short-term memory or your immediate memory could be called up by hypnosis. So they can make you regress back and you'll remember things that you've forgotten. So they're still stored there. And I threw this in because here's someone talking about being nervous. So something is making them fire their neurons. Something is making them think about something. And she's saying being nervous is a part of life. You're experiencing the full spectrum of emotions that you can feel as a human. And so I tell myself, this is living. The problem is I get nervous a lot. So some people are kind of lost in fears, phobias, past history. They divide up your long-term memory into explicit, where you can actually talk about it. So somebody asks you, what were you wearing when you went to school the first day of school? And you have to think back, and you remember whether you were wearing a uniform, or if you were wearing shorts, or your favorite t-shirt, things like that. And you'll be able to tell them about it. And then implicit are unconscious memories. So I have an old truck with a stick shift, and I only drive it about once every 6 months. So when I get out there, I think, oh my gosh, can I remember all the strokes that it takes to move that stick shift around? But I've been driving a stick shift for, 40 years. And so it's somewhere in there. I have a reflexive memory. I have the motor skills, and it comes back to me. I don't even have to sit there and think about it after just a second or two. I remember to push in the clutch, things like that. But now that I've gotten older, I drive automatic cars because they're easier on my knees, and they're easier to find. They're cheaper than stick shift cars. And of course, you got to have Calvin and Hobbes in there. So in the short term, it would make me happy to go outside and play. In the long term, it would make me happier to do well at school and become successful. But in the very long term, I know which will make better memories. And how do we forget things? Well, if you tear down the dendritic spines and remove little used synapses from your neural circuit, now you're not going to be sending action potentials down through that synapse and activating that neural circuit anymore. So you're going to forget. This is one of the hottest topics right now that old people my age are talking about. About 11% of the people over the age of 65 are going to have Alzheimer's. By the time you're 85, almost half the people will have Alzheimer's. Almost half. That's amazing. So they're going to have memory loss for recent events. Like I said, they may not remember what they had for lunch. They oftentimes become moody and combative. So they're a little bit frustrated at not being able to remember stuff. They lose the ability to talk, to walk, and to eat. And then eventually they end up dying, of course. There are early onset Alzheimer's. One of the smartest men that I ever worked with died early of early onset Alzheimer's. And he was the last person in the world I would have guessed that would have come down with that. So I was shocked to find out he had died long ago. And he was my age. So I was like, oh my goodness. They found that people who have Down syndrome have almost 100% early onset Alzheimer's. So they're thinking, well, maybe there's some sort of a genetic component in there. The people, if you look at their brain, you can't really diagnose. I mean, we have all kinds of tests that we know that we think that they have Alzheimer's, but to actually tell the person whether they really have Alzheimer's or not, you're going to have to go in and take a brain sample. And usually you don't do that until after they're dead. And you're going to find that they have amyloid plaques. They have neurofibrillary tangles in their brain. So basically they're filling their nerve cells up with this protein. And it's a normal protein, we all have it, but they start cutting it incorrectly. And it folds incorrectly, and it doesn't fit in the cell correctly. So it ends up killing the cells. Here's a picture of someone who has Alzheimer's. And as you see, that's not a normal nerve cell. That's not a normal neuron. So you've got these, you can clearly see these wads of protein filling up the cell body. Here's a picture of the amyloid plaque in there, and there's the neurofibrillary tangles all around there. And then there's some nerve cells that are not, see, there's a nice nerve cell, there's the nucleus, and look how much different this area looks. So if we could figure out a way to stop cutting the amyloid in the wrong place, then we wouldn't get the amyloid plaque, we wouldn't get the neurofibrillary tangles. Another thing that they see at autopsy, because these tangles kill the nerve cells, they kill the neurons, and they kill the associated nearby cells, the brain actually shrivels up. So you have atrophy of the folds and the cerebral cortex. Here's a picture of the brain where you can see that it is not a full, well-rounded brain. You can see areas where there's large gaps between the gyri. You have really wide sulci. I mentioned awakenings and Parkinson's disease. This is a result of dopamine-releasing neurons failing to release enough dopamine. So dopamine normally prevents excessive activity in the motor centers, but in the case of a person who has destroyed those dopamine-releasing neurons, or they are not releasing enough dopamine, you're going to have involuntary muscles. So one of the things that you have is facial rigidity. So they call it a flat affect. So they may be looking at you and they may say, "Oh, sweetie, I'm so happy to see you." And you look at their face and they're looking at you with just this blank look on their face. I remember my grandmother doing that to me. And I said, "Grandma doesn't like me." And they said, "No, honey, she can't smile. She's trying to really hard, but she can't. But she's saying with her words that she loves you." And then, of course, the slurred speech. So it's kind of hard to tell what they're actually saying. These people are not going to be able to do handwriting, that they're not going to be able to sign their checks or whatever. So they're going to have somebody help them. They're going to have trouble walking. They just can't get their muscles going correctly because they can't get this dopamine. And eventually, in the movie Awakening, they actually froze. So not just their face was rigid, but they also couldn't walk anymore. And I remember my grandmother had this pill rolling motion. You run your thumb across your first two fingers, and you just do it over and over and over. And it looks like you've got a pill that you're rolling with your thumb across your fingers. But of course, there's nothing there. It's just the motion of your hand shaking that causes the thumb to run across those fingers. So early on, they can give you dopamine, or they can give you something that your brain can turn into dopamine. So in the early stages, they can give you some treatments. Unfortunately, large amounts of dopamine are going to hurt your heart and they're going to hurt your liver. So the treatments that we have really bad side effects. They're wondering about how do people get Parkinson's. And I know from doing research on it, two different things that can cause Parkinson's. One of them is getting hit in the head repeatedly. So a lot of boxers that are in fights end up, like Muhammad Ali, with Parkinson's disease. In the case of my grandmother, she was in the flu of 1918, and she obviously didn't die, but she was left with Parkinson's. So they found a lot of the survivors of the flu of 1918 who ended up with Parkinson's disease. And I leave you with this something to think about. Your brain, with all of its neurons, doesn't feel pain. It understands pain. It can explain to you what you're feeling and why your toe hurts, why your finger hurts, why your broken bone hurts. It can tell you about things like that, but it itself doesn't feel pain. So if they have to do surgery on you, on your brain, they'll not put you to sleep. They'll leave you awake. They'll numb the tissue. Obviously, when they cut your scalp, it's going to hurt. So they're going to numb that tissue. And it's amazing to me that they could be playing around with my brain and me talking to them. And they say, well, what do you think about this? And I'm telling them what I think about that. So that's amazing. So you say, well, no, wait a minute. No, I know people get headaches, so your brain must hurt. Well, what you're feeling when you have a headache is blood vessel spasms. So let that sink in.